Hello, everyone. Good evening. Thank you for joining us here tonight. My name is Henry Scowcroft. I'm Head of Communications here at Alzheimer's Research UK. It's my absolute pleasure to welcome you to be hosting tonight's discussion. Before we get started this evening, I'll just go over a little bit of housekeeping. During the event, you're welcome to switch on the subtitles using the CC button at the bottom of your screen. These subtitles are auto-generated. They're not 100% accurate during the live event, but we'll be editing them so they are correct on the event recording, on which point we will be sending you a link to the event recording afterwards, which will be available on our website and our YouTube channel, probably in the next week or very early in the new year. Tonight we'll be answering a mixture of pre-submitted questions, which we've used to help shape the discussion, but also live questions as well. If you would like to ask a live question, please do click the Q&A button at the bottom of your screen, which will bring up the Q&A box where you can type your question. We've received a whole load of fantastic questions ahead of the event, and we're going to be weaving those into the discussion as we go. We're now going to ask you a few poll questions because the nature of this event means we can't see you, but we would like to know a little bit about who you are and why you're here watching this event. So we've got a few questions which you should now be able to see on your screen. First up, how would you rate your knowledge of dementia research? From answers ranging from very high to very low, or I don't know. Second question is, which best describes your reasons for attending the event? There's a variety of options there, and just be really good to know if this is your first event with us, or whether you've been to several of these beforehand. So just give you a bit of time on these questions. Give you a couple more minutes to do that. Here we go. So we can see from the answers that most of you rate yourselves as having an average knowledge of dementia research, with some high, some low. Many of you are here because you have dementia, or a friend or family has dementia, or are a carer. A few people whose jobs are related to dementia, and there's some of you here that don't have a personal connection but want to find out more. We've got a almost 50-50 split on first timers and repeat visitors. So welcome. Thank you all for joining in. So I will now introduce the panel to you for this, our Conversation at Christmas event. First up, we have Paresh Malhotra from Imperial College London.
Hi, Henry.
Hi, Paresh. Would you like to introduce yourself to the audience and say a bit about your background and what you do day to day?
Yeah, hi everyone. So my name is Paresh Malhotra. I'm a clinical neurologist at Imperial College. I do 50% clinical work, seeing people living with dementia, making diagnoses, treating them, and then 50% of my work is research, working particularly with Alzheimer's disease and vascular dementia.
Thanks very much. And I'd now like to introduce our second panelist, Fiona McLean, Dr. Fiona McLean from University of Dundee.
Hi there, my name's Dr. Fiona McLean and I am an Alzheimer's Research UK Research Fellow, and I'm based at the School of Medicine at the University of Dundee, and I am 100% research. So I spend all my day in the lab, as well as working with students that come through the university who want to learn about the brain and neurodegenerative diseases like Alzheimer's disease. And yeah, that's me.
Brilliant. Welcome both. I'm Henry Scowcroft. I'm Head of Communications at Alzheimer's Research UK. We are the UK's leading dementia research charity, and we are working on all fronts against dementia to find ways to treat, diagnose, and prevent it, with the ultimate vision of a cure, really, to try and find ways to end the fear, the harm, and the heartbreak of dementia. Last year, we committed £23 million towards dementia research. And we really feel like we're standing now as a community at a tipping point, where we feel like there are new treatments coming through. We're starting to revolutionise the way the disease is diagnosed, and we're really starting to understand more about the factors that influence dementia risk to be able to maybe prevent it as well. So a huge amount of excitement and promise and hope going on in the field of dementia research. What we want to talk to you about tonight, something called amyloid. Amyloid is one of the hallmark proteins in Alzheimer's disease. The first major discovery, really, about the biology of Alzheimer's disease focused on the discovery of this protein in the brains of people who have Alzheimer's disease. It seems to build up as the disease progresses, and it forms things called plaques, which surround nerve cells in the brain. But there's still so much to know about amyloid, and what we're going to be talking about tonight is exactly that. What do we know about amyloid? What's that opening the door to in terms of new treatments and new ways to diagnose Alzheimer's? But also, what do we need? What else do we need to know about the disease and how it causes dementia? So, to start with, we'll just talk a bit about the history of amyloid, how it was first discovered, and Fiona, can I bring you in to talk about the biology and the science behind amyloid and what it is?
Sure. So amyloid plaques, as you mentioned, are one of the hallmark pathologies that are seen in the disease, particularly in Alzheimer's disease, but not limited to Alzheimer's. It does crop up in other dementia-related conditions as well. And these amyloid plaques were actually first described a really long time ago, in actually the 1800s, towards the end of the 1800s. And one of the first two scientists that mentioned these plaques in their work were Paul Bloch and George Manescu. And even back then, they were linked with dementia. And then they are now known, as I said, to be a hallmark of pathology. And they are very much related with Alzheimer's disease. But where does it come from? And this is what a lot of the work in the last, I would say, 50 years has been on, is where do these plaques come from? Well, amyloid is produced from something called amyloid precursor protein. It's sometimes referred to as APP for short. So you might be thinking, well, if we know where amyloid comes from, then why can't we just get rid of APP and then that will get rid of amyloid? But actually, APP has some really essential functions in cells, and we're still learning about what those are. But it looks like it's a very hard-working thing, and it's actually responsible for keeping cells alive and healthy. So we can't just get rid of APP. But we do know that APP can get chopped up into smaller bits within the cell. And those smaller bits also have important jobs. But sometimes when APP gets chopped up in a certain way, it can produce a fragment, which is called an A-beta peptide or A-beta peptides. And it's these types of fragments that can get stuck together and form what we now know as amyloid plaques. But what we still are working on is trying to work out if these plaques cause dementia, or are they a symptom of dementia and something else happens before these plaques are formed. And that's what a lot of the work that ARIA UK funds is trying to find out.
Thank you. And I guess the way amyloid appears in the brain and what it does, do we know much about how it affects cells and its surroundings?
So there's a few camps of thought. There's thoughts that these fragments could be quite toxic to cells. For a long time, it was kind of thought that these plaques were just found in and about what we call neurons, which are the main nerve cells of the brain. And neurons are, they have a sort of body of the cell, and then they have projections that come off, and these projections actually touch and talk to each other. And it's by the communication between these cells that memories are formed. So that all kind of made sense, that if you got plaques in the way of that, or plaques that would break down those connections, that could lead to memory loss. But we now know that plaques can be found associated with other cell types. So a lot of my own work is actually looking at how amyloid plaques are associated with brain blood vessels and a type of cell which makes up the brain blood vessel called endothelial cells. So we're still trying to work out what part of the amyloid process is toxic. So is it these plaques, or is it the fragments early on? And there's also a lot of thought that these plaques might actually be being formed to stick to other toxic things that might be building up in the brain as a result of other things. And actually these plaques might be attempting to be protective. So you can almost think of them as dustbins. They're maybe trying to collect the rubbish and keep it together rather than let it spread around the brain. So that's what we think. We're still trying to unpick exactly what they're doing and why they're there. But we are getting closer to finding out.
And some of our viewers may have come across the name of another protein called tau. And I just wondered if you could talk a little bit about how amyloid and tau are related, and how they may also have heard something called the amyloid cascade hypothesis, or the amyloid hypothesis. And just talk a little bit about amyloid and tau and how that all fits together in Alzheimer's disease.
Yeah, so the amyloid cascade hypothesis is kind of what I was describing before about APP, so that amyloid precursor protein getting chopped up and then turning into fragments, which then form what we call fibrils, which are sort of longer versions of these fragments stuck together, and then they form these messy sort of plaques, which are all kind of like a bundle. Tau is the other hallmark pathology of Alzheimer's disease. And I've got to say, I've done a lot less work on tau, but where my interest has come along with tau is when tau is working in a cell, it associates with something called microtubules in the cells. And these microtubules are really important for maintaining the structure of cells. And microtubules are, I always think of them as almost like train tracks in the cells. So things will get shuttled up and down them. For example, maybe nutrients which are in the middle of the cell might need to go out to those long projections that I was talking about earlier that make those connections with each other. So the microtubules be responsible for that. And tau is part of that function. But what happens in Alzheimer's is something called hyperphosphorylation, which basically means that they get bits added to them that then change the way they can work, and it just stops them working properly. And it's almost a similar thing in that these bits that get added to tau then cause it to become what we call a tangle. So we call them tau tangles. And as I said, both these pathologies are hallmarks of Alzheimer's disease.
Fantastic, thank you. So we've got amyloid building up outside the cells, and then a bit later on, or some other point of the disease process, the tau tangles form inside the cells, which seem to be and this whole process seems to be associated and happen at the same time as the nerve cells are dying in Alzheimer's disease. But I'm right in thinking that whole process, we've observed that many times in samples from thousands and thousands of people, but we still just don't quite understand why the amyloid builds up, how it causes the tau to build up, and how that all is associated with the death of the nerve cells, which leads to the symptoms.
And there is quite a body of evidence now emerging that actually there is the beginnings of cell death that happen before we see these pathologies.
Right.
So a lot of my work is actually, so the work that Alzheimer's Research UK fund is actually looking at what I call pre-pathology. So before this happens, what is changing, to try and understand what might be leading to these pathologies. And for us moving forward, looking for treatments, that's really important, because it's very hard to treat something when it's at such a severe stage where we've got these pathologies already present. So we want to find out what's happening before that, so we can find interventions earlier, so that we can prevent the subsequent cell death that happens.
We've got a question from a viewer. Gregory Peters asks, when does the brain begin to shrink like a cauliflower drying out? Is that separate to the plaque development? I think from what you're saying is that we don't quite know, but they're related somehow, but it's still a bit of a question.
Yeah, I think typically my colleagues who do lots of brain scanning are much better at understanding that, but the brain volume loss is due to cell death, and the cell death is happening later on in the disease progress. But like I say, what we really need in research is to find out what's happening before we get to that stage, because it's very hard to replace, bits of a brain. But if we can stop it, shrinking in the first place, that's our goal, because then you're preserving your brain.
Thank you. I think that's a great point to bring in Paresh, Professor Paresh Malhotra. So Paresh, we've known about amyloid for a while, and it was sort of first in the queue when it came to trying to develop treatments for Alzheimer's disease. And we've all seen the headlines over the last year or so about these new drugs, lecanemab and donanemab, which are the first drugs that seem to be able to slow down the processes that we've been talking about. Paresh, can you talk a bit about those drugs and how they're related to this amyloid hypothesis and how they work?
Yeah, sure. So, I mean, the aim, the target of lots of people's research over many years, so more than a decade, more than a couple of decades really, has been trying to remove amyloid from the brain to see if that will help people living with Alzheimer's disease. And so many years ago, people developed a vaccine, and that was seen eventually to show that you could remove amyloid using the vaccine approach.
Could you just explain how a vaccine, which most of us think about in terms of infectious diseases, could you just explain how a vaccine might remove a protein from the brain?
So, like, you're trying to get the immune system to do the job of mopping up the amyloid, basically, and by showing it a version of that amyloid, then you're priming it really to attack the amyloid within the brain itself, within the nervous system. And so that has been shown to affect amyloid, but unfortunately it was also associated with significant side effects in quite a proportion of people given the vaccine. So that trial was stopped. But interestingly, people are getting more interested in vaccines again. But in the meantime, people have used sort of the developing technology we have with antibody-based drugs. So in rheumatology, for arthritis, for cancers, people are modifying antibodies, modifying the sort of weapons that the immune system has to try to be able to attack a specific target. And in this case, it's amyloid. And about 5 or 6 years ago now, a drug called aducanumab in a sort of early study was shown that the more you gave of aducanumab, the more amyloid was removed from the brain. And we have amyloid so-called PET scans where we can see the amount of amyloid in the brain and where it is. And there was a very nice picture in that paper where you could see with increasing doses of this drug aducanumab, more and more amyloid was being removed. And since then, more trials have taken place of different compounds, including aducanumab, but more recently, these drugs, lecanemab and donanemab, all of them with the ab at the end to indicate that they're an antibody, a monoclonal antibody. And these two recent ones, lecanemab this time last year, the results were announced, and donanemab this summer. The interesting thing that's been found, and the thing that's got people all over the world very excited, is the fact that these drugs remove the amyloid and they also appear to affect symptoms. And it's that combination of appearing to work on the fundamental basis of the disease, a core part of it, and changing symptoms that has got researchers and clinicians very, very excited. And to be honest, there's some controversy about it, But all researchers, I think, that I know are very excited about this breakthrough. And then the controversy mainly lies within discussion of how clinically important or effective these drugs are going to be for patients soon and now, if you see what I mean. So that's the current state of affairs with those two drugs.
And when you say clinically important and how they affect symptoms, I think many people will be familiar with the idea of the current drugs that are available for diseases like Alzheimer's disease, which can kind of influence symptoms. How do these drugs influence symptoms, and what's the discussion around clinic effectiveness, and what do we know from the data we have so far from these trials?
So this is a very contentious topic. So I'm trying to think about every word I say before I say it. But first of all, they don't improve symptoms. They make symptoms less severe than they would be. So that's a very important thing to note. The second thing is that initially, at least, they don't affect the symptoms as much as the drugs we already have. So they're not hugely effective straight away. But the really important thing about them is that from the effects that have been observed over treating people for a year and a half, 18 months, there seems to be some signal showing that they slow the progression compared to people who are on a placebo. And this is also quite contentious because it's 18 months and everybody would like to know what happens for longer. But in the data, in the results we have for the time that people took these drugs, there seems to be a separation. So if we have a line going downwards for the people who are on placebo, the line for the people on the drugs seems to be moving away. And there's argument, will it continue to move away? And so will the underlying disease be modified by these treatments? And that's what all the excitement's about.
But the clear message that I take away from this is that we've understood the underlying biology of the disease to a degree. We've designed a treatment that can try and modulate that biology, and what we're seeing is an impact on the rate at which the disease progresses in a way that tells us that this is a disease that it looks like we can finally, after a lot of trying and a lot of effort and decades of research, we can target the biology of this disease. And that, to me, strikes me as a moment that it pushes the door open.
Yeah, and that's why I wanted to say that all researchers I know are very excited about this breakthrough, and I think that they realize how much it matters. And at the conference last year in San Francisco, where they announced the lecanemab results, there was literally cheering throughout the room. So it makes a big difference, and it gives a lot of people a lot of hope.
I think that's the message from this is that this is the door is open, and we know we can push it open further. But also just to temper that with the knowledge that there's a long way to go to really push it fully open. It's ajar, and I think we can talk a bit about how we push that door further open. But we've got a couple of questions from the audience. Going back to the presence of amyloid, how early can it be detected? How early can it be picked up? And we can talk about using current tests now, and we can talk a bit about how we might do that in the future. But when would you start to be able to see amyloid appear? And then a related question, just to wrap them both up, is amyloid found in people who don't have dementia?
So both great questions, and Fiona, just interrupt if I say anything that you disagree with or what you want to add to. But first of all, that it can be detected more than 10 years before symptoms start. So that's very important. So some people would see it as a trigger to what happens next, that the first thing is being able to spot this amyloid, and then much later the symptoms. And someone mentioned the brain shrinking, and the brain shrinking can be at least 10 years after amyloid is first detected. It can be detected by the PET scans, which involved a radioactive injection of a material that will stick to the amyloid in the brain, or it can be detected using a lumbar puncture, where we take a small amount of fluid from the base of the spine and analyze that, or it will soon probably be able to be detected, or can be detected, with blood tests. And those blood tests are just undergoing validation and sort of preparation for how they'd be used. So that's the answer to the first question. Is that, Fiona, did you want to add anything to that?
No, I think you've sort of covered it all just about the blood test. That's been a real goal of researchers for a very long time because, as you'll know, going to the doctor for other kind of tests, taking the blood's the easiest. You can get it done at your GP. So that's been a real goal, and Alzheimer's Research UK have funded projects that have been trying to reach that goal. So for those to come through and be really available to people is really important. Just something to talk on the treatments that have come out is about, maybe I'll touch on this a little bit later, but about the availability and how those are delivered. And one thing we really need to think about is the infrastructure of the NHS to deliver treatments coming through. So maybe we'll move on to that later, but the blood tests would be are something which would be more available. So yeah.
And just to talk talking about the blood tests, we are soon going to be announcing that the successful applicants for a very big research grant that we're going to be jointly funding with Alzheimer's Society and the National Institute of Health Research to really put these blood tests through their paces in the NHS. We know that they don't actually. My understanding is they don't detect amyloid. They detect tau, I think, which seems to be a bit more tightly linked.
They detect tau. Well, they detect a substance called p-tau, is the sort of the most exciting one, which weirdly is a very good indicator of amyloid.
Yeah. Which is another conundrum for another day. But the research project will be looking at the tests that have been developed and to be able to work with high accuracy in a research setting, but trying to understand how they, as Fiona says, how they work in the context of the NHS, how they can be integrated into usual care, how they work with across really diverse populations. That's the thing we really need to crack, and that research is going to get underway very soon. Really going to be excited to talk more about that next year. I think it's probably good. That's a good pivot point to talk now about the challenges of these treatments and what they mean for what they could mean. Just to get so we're all on the same page in terms of where they are. The trials have shown that there is a positive effect. The drugs are now being looked at by the regulators, who will make a decision on whether they are safe and effective, and then they will need to be looked at in terms of their cost effectiveness by the NICE, which is the organisation that looks at cost effectiveness. Paresh, how do you see that playing out next year, or rather, how does that, when you are talking to people who come to your clinic and asking, where are these treatments, when are they going to be available? Talk a bit about the conversations you're having now, because there's been a lot of media coverage of this. How's this playing into people's perceptions?
So first of all, I think stepping back a little bit from the excitement we were talking about, or I was mentioning earlier, I think it's very important to discuss what they mean for individual patients and what they mean to them. And so, for instance, these drugs aren't for everyone with every form of dementia. They're not for everyone with Alzheimer's disease. They really have only been shown to have these effects at the earliest stages, or what this stage called mild cognitive impairment, and then the stage of mild dementia. So for a lot of people, they've not been tried in that group of people with moderate disease, and it's thought that they would not help. And they are associated with side effects that we can come to later. That said, what I say to people is just about what you said, that the MHRA, the Medicine Regulating Authority, is looking at things now. Even though NICE don't know what the MHRA's decision will be, they are already looking at this now. And then lots and lots of people are thinking about the questions you mentioned, you and Fiona both, about how this would fit within our NHS infrastructure. And it's, I do a clinic every Monday morning, and we have a discussion after clinic. And almost every week in our discussion, which is about patients and about their management, we're starting to think that, what would things look like if treatments were available? What would we do? What more would we need to do for that patient to make the diagnosis or to confirm it? And what might exclude them from treatment, for instance? And we my colleague Richard Perry has run some of these trials here on the 10th floor at Charing Cross Hospital. So he has experience of the side effects, but also of the stuff that Fiona mentioned, you know, that infrastructure that requires people to have infusions, intravenous infusions every couple of weeks, every month.
Because this isn't a tablet you can take at home, is it? You need to go into hospital and have a intravenous infusion.
Yeah, so it's there. I mean, it's not to be taken lightly, and it's not to be discussed without considering the side effects and what we mentioned earlier about the clinical effectiveness and how much there is. So I think being sort of, what's the word, being very honest with patients and their families and trying to be absolutely clear about the risks and benefits will have to come whatever, well, if the regulators make decisions to make these drugs available.
And I think you mentioned side effects, and I think it would be remiss not to cover the side effects in this discussion. Can you just talk a little bit, first Paresh, in terms of what the side effects are, and then perhaps maybe Fiona, if you could talk a bit about how they might be caused by these drugs, as much as we know, which I gather is not a huge amount. So Paresh, what are the side effects of these drugs?
So the main one the main type of side effect that has been observed is this thing called ARIA, which relates to things that we see on the scan that in the majority of people aren't associated with symptoms. So we have two types of ARIA. ARIA E, which is edema, which is swelling in an area of the brain after people have been given the drug, or ARIA H, which is hemorrhage, so bleeding in the brain. And both of these can be without symptoms and have been observed on the scans, and each of the trial protocols has instructions for what to do if a certain amount of ARIA has been observed. But in a smaller proportion of patients, you know, around 5 or 6% roughly, then these symptoms are much more likely to be symptomatic and associated with the problems that you might see with brain swelling or brain hemorrhage. And in a much smaller proportion, they've been very serious and sometimes fatal. So these are not things to be ignored. And I think, as you said, it would be remiss of us not to mention them in a conversation about treatments.
Thanks. And Fiona, what do we know about how removing amyloid might lead to ARIA?
Yeah, so a lot of my own work is understanding, as I mentioned before, these amyloid plaques and how they associate with the brain blood vessels. And sometimes that's referred to as CAA, or cerebral amyloid angiopathy. But even though that is its own thing, in most Alzheimer's cases, so in a huge proportion of them, you actually get CAA. Correct me if I'm wrong, Paresh. But yeah, you see this association of amyloid with the brain blood vessels. So I think what we think is going on is when this amyloid is being removed, it's maybe being pulled out, and that might be creating basically sort of little holes in the blood vessels. And something that I do a lot of work on is something called the blood-brain barrier. So the blood-brain barrier sits in the blood-brain vessels, and it sits between the blood and the brain nerve cells. And it controls, when it's working properly, it controls very well what gets into the brain and also what gets out. So I like to think of it as a bouncer on a nightclub door. So it emits all the good things and it doesn't let any riffraff in. And if there's any trouble in the brain, it'll kick it out as well. And we know that in Alzheimer's, actually, this barrier, it stops working properly. But back to what we think might be happening. So yeah, we think that maybe these drugs, when they're pulling out the amyloid, it might be creating little holes in this barrier that might then be leading to these sort of what I would call a microbleeder, as Paresh was talking about, these sort of hemorrhages. So I think there's a lot of work to be done to understand how these drugs are removing amyloid. There might be then the opportunity to refine it, to maybe be able to still remove the amyloid without causing these side effects.
So we've got some drugs. They're new drugs. They're coming through. They're going to be a challenge for the healthcare system, but they are the first drugs that make an effect, on the disease process itself. I think that's almost the moment we want to move on to talk about, well, what happens next. But I've got some questions, so I just want to get to the questions box and see if we can answer some of them. We didn't quite cover the answer of, is amyloid found in the brain
Oh,
if
yeah.
you don't have
Yes.
dementia? So let's just
Yes.
skip back to that point. Who would like to take that one on?
I think Paresh is probably I think we probably agree on it, but Paresh is a clinician, so he's probably seen more of it.
The answer is yes, basically. And so especially with increasing age. So in over 80s, up to 50% of people, if you look for amyloid, have it there without, up to 50% of people without symptoms. And so understanding what's stopping those people from getting symptoms, is it that they just haven't developed them yet? Or is it that they have some resilience, and we know a little bit about resilience, but certainly not enough? These are still key questions to be answered. But amyloid itself, if it's without any tau present, is unlikely to lead to symptoms, whereas it's the tau and where it is in the brain that seems to be much more closely associated with symptoms.
And this is what is maybe leading scientists to think that amyloid might be more of a symptom or something that is trying to gather up sort of things that are causing trouble in the brain, toxic things. This is what's leading scientists to think that maybe amyloid actually has a, dare I say, protective role in the brain.
Thank you. And we've got another question. We've talked a bit about amyloid PET scans that detect amyloid. We have a question about whether an MRI scan would show anything. Paresh, probably one for you there.
Yeah, so we use MRI scans a lot, and they show shrinkage as people have symptoms. They're unlikely to show shrinkage before people have symptoms, but sometimes they do. But a couple of important things is, one thing is that shrinkage can be caused by other disease processes. Another thing is an MRI is just a picture, it's a picture at one point in time. And so it doesn't tell us what disease process is taking place, but it doesn't tell us when that shrinkage occurred, or whether someone's brain was always a bit like that, if you like, if that shrinkage happened 30 years ago or something. And so 2 MRI scans 18 months apart, for instance, is much more useful to tell us about progression and changing of shrinkage, but that still won't tell us exactly what the underlying cause is and whether it's Alzheimer's disease. So they are very useful, and they're amazing technology to tell us about the structure of the brain.
Great, thank you. Let's talk a little bit now. Before we pivot to the future, it's a very quick thing about how these drugs are changing the conversations. And Paresh, you talked about the conversations you're having about preparation for these drugs, but in terms of how this might affect people's perceptions of dementia. And I think we know from our own work, and we publish this thing called the Dementia Attitudes Monitor every couple of years, and it shows that there's a substantial part of the population think that dementia is just a natural consequence of aging, whereas what these drugs seem to be telling us is that no, these are specific diseases that cause dementia. Could you talk a bit about stigma and about perceptions of dementia around this whole phenomenon?
Absolutely. So I think even in the last 10 years, the amount of stigma about diagnosis has changed. So when I started as a consultant, people were much less happy to tell their friends or perhaps even distant family about their diagnosis. And perhaps even more importantly than that, they started to isolate themselves a bit more and would not go out as much because they found it difficult to cope unless people made sort of special consideration for them. A noisy restaurant is tricky if you can't quite follow the thread of what someone's saying. It's difficult to recall everything that might happen to someone that if you only see them once a year at a Christmas party. And so unless you're able to tell people about what the problem is, whether it's memory or whether it's being able to focus in a crowded room, things get more difficult. And then over the last 10 years, people are a lot happier to let people know that they've got a diagnosis of Alzheimer's disease or they've got cognitive impairment, and discuss these things and say, oh, I've started treatment, I'm in a treatment trial. And I think that's made a very big difference. And one thing that I've noticed is, I started at medical school about 30 years ago, and in that time, I've seen the treatment of some diseases absolutely revolutionized. So HIV is a very good example. Stroke is another very good example. Many cancers are very good examples of things we thought of as in very different ways 20 or 30 years ago. We think of as very treatable conditions that hopefully are much more of a nuisance than a major life-altering diagnosis.
So I think that really neatly segues into the last section of this, is where we take where we are now and look into the future and think where might we be. And I think one of the things that's come out from the conversation so far is that while amyloid seems to play a very important role as a hallmark of the disease, and removing it seems to make some difference to the progression of the disease, we need to do so much more than that. And I think we've sort of skirted around the issue a bit, but particularly Fiona, with some of your work on the blood-brain barrier and in terms of the metabolism in the brain and the immune system in the brain, there's so much else going on here that could be some stuff we target in terms of the next generation of drugs. And that's something we're very passionate about at Alzheimer's Research UK, in terms of trying to broaden out the range of potential treatments beyond that. Fiona, could you talk a bit about your own work and where you see some potential avenues for new treatments opening up?
Of course. So this is where I say I actually didn't start out as an Alzheimer's researcher. I started out as a type 2 diabetes researcher, and we were really interested about the effect of type 2 diabetes on the brain. And the reason that I ended up getting linked with Alzheimer's disease is because if you have type 2 diabetes, you're about twice as likely to go on and develop a dementia-related condition like Alzheimer's disease. So there's a really clear bit of evidence there that there's an association between type 2 diabetes, which involves usually being obese and having high blood sugar levels, and then developing problems in the brain that then end up with dementia. So a lot of my work has been trying to find out why that is. And Alzheimer's Research UK have funded my research, which is based on a foundation of actually understanding that having type 2 diabetes and that obesity and high blood sugar levels that we were talking about, having that actually results in the blood-brain barrier changing the way it works and breaking down, and actually changing the way that things are moved across the brain, both in and out. So that's actually where my research comes from. And also, really importantly, is understanding not just what's happening in the brain, but what's happening in the periphery in Alzheimer's as well, because our brain isn't an isolated organ. It sits in our body. It's talking to all our organs. In fact, it's the control center of all our organs. And so I think there's been a real gap in the research of understanding what's happening in the rest of the body alongside these changes that we see in the brain. And yeah, that I'm about a year and a bit into that project, and I've just started to get some data coming through. So hopefully we start to understand better about this whole system, I like to call it. So brain, organs, whole system, what's going on in Alzheimer's.
Thank you.
And should probably actually talk about lifestyle. That's what I was going to say next. So one thing which we've been talking about is treatments coming through. Before that was announced, my project started, and we actually come from a place of looking at what you can do for yourself throughout your life to prevent getting dementia, prevent getting Alzheimer's disease. And like I say, there's this really strong correlation between type 2 diabetes and developing Alzheimer's. So a lot of the things that actually you can do to look after your brain are the same things that you can do to look after your heart. And I think there's been great campaigns around heart health and, eating foods that are low in saturated fat, not eating too much refined sugars. And those are actually kind of the same rules as you should think for your brain. You don't want to be eating unhealthy foods. And the other really good thing is exercise. I think I'm yet to read about a study where exercise hasn't resulted in a positive effect. Maybe Paresh can comment, but I don't think I've ever seen. We tried exercise and it didn't help our brains. So that's really important. So getting outside, moving, and also I think this was kind of touched on, is staying social. So there's a lot of research around if people become isolated their conditions can get worse more rapidly. So it's really important to stay social. That can be really, really difficult when a diagnosis is received, because it's scary and it's worrying, and it can be worrying to tell friends and family. But actually, it's really important to share that. It's important to try and, you know, still be included as much as possible, to still stay social, and to try and keep independence, because that is actually probably going to help your brain sort of retain some of its ability to think and to focus as well. So yes, that's my take-home message: is what's good for your heart is good for your brain.
An excellent message. Paresh, in terms of your perspective on where we're going with all of this and what's needed, some thoughts from you and the work you're doing and where you see things in 5-10 years' time.
So I think what I'm about to say follows on from the fact that I do a lot of a mishmash of work, really, that some of it related to prevention in the same way that Fiona has discussed. And also, I'm very interested in the sort of interaction between different organs in the brain. And for instance, a lot of the patients that I see, they haven't just, in inverted commas, got Alzheimer's disease. They have diabetes, they have high blood pressure, they may have some chronic kidney impairment, and these things interact. So thinking about how the organs interact and how they contribute to worsening of the overall condition, I think is very important. And thinking about prevention. But in the meantime, whilst we're thinking about these things, hundreds of thousands more people are getting dementia and getting Alzheimer's disease, and thinking about how we treat them, both from a disease modifying point of view. So as you've said, and about that door being ajar, if we stop now and stop trying, we're really not going to get very far at all. So we have to keep developing things. And similar to the way we do treat HIV, for instance, thinking about having a cocktail of medications and thinking about which medications might be useful at different points in someone's trajectory. So maybe there are different approaches to use if someone has amyloid, but they only have a small amount of tau, which is what seems to be the case for donanemab, at least. And then there may be medications that may be much more helpful later, and not just medications, but different treatment approaches. So people are pioneering work in things like brain stimulation, in focused ultrasound approaches, all sorts of things that at the moment we think of as slightly outlandish, and then as soon as they work, we'll be thinking, oh, why didn't we ever realize that that would be the key? So I think having a sort of approach across the board, trying to address these Alzheimer's in every way we can, is going to be more successful.
And that's what I hear really strongly when I talk to the folks in our research funding directorate about how making sure that we try and broaden the number of approaches that are going on here. And I've heard talk of treatments that target the immune system to try and increase the way that the cells of the immune system within the brain clear out the stuff that shouldn't be there, but also that the immune system can sometimes get stuck in the wrong state, so start doing bad things within the brain. And you know, seen some research that suggests that it attacks the synapses, the connections between nerve cells and everything. So and you can see in other fields, like in cancer, where you've got immunotherapies that change the behavior of the immune system to point it in the direction we want it to go in, and not in the unhelpful direction. It feels like there's so much out there in terms of the need for more research, the need for more drug discovery, the need for more clinical trials. I mean, we haven't really talked about clinical trials in the UK, but we need many more of them to be running. And these are all things that Alzheimer's Research UK, we're really, really passionate about, you know, moving forward and trying to increase the diversity and the breadth of the targets beyond drugs that simply remove amyloid from the brain, while recognizing that that's what we have in front of us at the moment, and they are effective for some people in some circumstances. They appear to be from the trial data. We need to wait and see what the regulators say. But yeah, I think we're getting towards the end of the session now, so I think we'll wrap up the Q&A bit now. I'm sorry we didn't get to more of your questions. But I wanted to sort of ask the two of you just to reflect for a couple of minutes each on where you see us as 2023 comes to a close. We've seen the headlines. We're looking at next year. What do you hope to see over the next year and over the next few years happen? Paresh, I'll go to you first.
So coming back to clinical trials, I started a clinical trial of what we call a repurposed agent, so something we use for another condition, which is a drug that's used in ADHD, attention deficit hyperactivity disorder. And I started that almost 10 years ago now, and we're due to finish next year. So the first thing is to look at the results of that and to see. And obviously, you know, the odds are against anything suddenly working, but if we see any effect on symptoms, that would be, you know, a really amazing thing to see. And then the same across the board really is to see the results of the trials that are going on now. We've mentioned amyloid a lot, but to see what happens with bigger trials of agents that remove tau or affect tau. There's a lot of stuff coming through now, and because of the excitement about the amyloid trials, people, not just scientists, but also companies that invest in drugs, are putting more effort into things again. So trying to recruit people, trying to make sure that taking part in research is seen as the norm rather than anything else. Just a big boost in research in general and trials in particular over the next year would be amazing.
Brilliant. Thank you. Fiona.
I think in my own research, so like I say, I'd call myself a fundamental researcher. So at the other end to Paresh, in my own research, I'll be getting some data through looking at what I was talking about earlier, which is the pre-pathology stages. So I really want to try and unpick what's happening before that amyloid pathology really sort of becomes a real problem. And also from more of my diabetes focused work, we have potentially something that we think might be causing an uptick in APP, that amyloid precursor protein. So we're hoping to sort of get some more funding in to look at that, because we think we found an association between, like a biological association between type 2 diabetes and Alzheimer's disease on a molecular level. But talking about funding, I think this is where I'll end my hopes, is that I really, really hope that we see more funding. And Alzheimer's Research UK are absolutely fantastic. I wouldn't be doing Alzheimer's research without their support. They fully fund the research that happens in my lab. They cover my salary so that I can do this job. And a year and a bit ago, if I hadn't got that fellowship, I probably wouldn't be an Alzheimer's disease researcher. So what I hope to see is actually not just so much charity funding being depended on. I'd like to see more government funding. I'd like to see a real commitment from our government towards really going for it, because, you know, this is a disease. Dementia is not normal aging, and if it is a disease, then we can cure it if we can find those cures. But to do that, we need the funding. So yes, I really hope that the government, whatever that government is in the future, really commits and really delivers that funding.
Thank you, Fiona. I think we all share those hopes and those sentiments. I think it's time to wrap up the conversation. Thank you so much to both of you. I think just to summarize what we've talked about today, I think we've talked about amyloid, this protein that appears in the brain of people with Alzheimer's, and we've talked about how we know it's deeply enmeshed in how the disease develops, but there's still so much we don't know about it. We've got these drugs that can remove amyloid from the brain that start to show the first signs that we can shift the dial against this disease. That tells us that we're on the, you know, research will take us there. We can do something about this. We've heard that these drugs are going to be challenging to deliver within the NHS. The NHS is going to have to think about how to bring them in and how to. And we know that, you know, there's definitely thinking going on in that regard within the upper echelons of the health service, but. They're going to be a big disease, a big paradigm shift in how we look at this disease, but that can also help shift the stigma around it, because actually something that can be treated means something that can be much more kind of openly talked about and not hiding away in the shadows. So there's some you know, two sides of the coin there, and we know these treatments they've got to pass the regulators, they've got to pass through NICE. We hope those decisions happen as quick a time frame as they can, Just seen a question that we have had about what realistically are we looking at in time frame. Next year or the year after seems to be the educated guess on this one, but there's a lot of unknowns there. But yeah, it feels like these drugs are just the beginning. That, you know, for a cure, we're going to need drugs that target other pathways. We're going to have to understand much more about the disease. It feels like such an optimistic time, but a time of optimistic realism where we know what the challenges are ahead. We've got a lot of work to do, but we know that we're heading in the right direction. So I think I just want to say a big thank you to Paresh and Fiona for a really fascinating discussion. Thank you so much. Have a wonderful Christmas, both of you. Keep in touch. Let us know how you get on, and let us know if you make any massive breakthroughs that we can come and talk to you about in a future lab notes. But yeah, thank you very much. I'll say goodbye to you both now. We've just got a couple more bits of housekeeping and wrap up to do, but happy Christmas and thank you so much.
Thank you.
Thanks. Bye.
So before we go, just a couple more little bits to cover, a couple more slides to share with you, and a couple of poll questions. We'd love your answer to give us some insight on how you found today's discussion. We've got an end of event feedback poll. So we'd just love to hear from you about what you thought of the content of the talks, whether you would recommend this kind of event to family and friends. I hope the answer is yes. And we'd like to know if you've learned something new about dementia research today. We hope you have. I hope it's been a stimulating discussion. I thought Paresh and Fiona made some fantastic points and learnt a lot myself over the course of the discussion. But yeah, if you could pop your answers in the poll, I'll share them with you just as soon as they come through. So the feedback is, yeah, happily 95% of you thought it was about right, which is great. More than half of you, no. A very high percentage of you, I can't do the maths, but would agree or strongly agree that you would recommend this, which is also great to hear. And fantastic that almost all of you say that you've learned something new about dementia research. So that's really great to hear. Thank you, and I'm glad that we were helpful in our discussions this evening. Just a couple of final bits before we go. I know there are a lot of questions that we weren't able to get to during the event, and that you might need some more personal information or guidance. Our dementia research info line is here for exactly that reason. They can answer questions and signpost you to other sources of information and support. So please do get in touch with the team that run that line. You might also be interested to know, and we've talked a bit about clinical trials and taking part in research and the importance of the participation that drives all of these discoveries. There is a service called Join Dementia Research, which can match you to studies that are taking place across the UK, clinical trials, but also studies trying to find out more about the diseases that cause dementia themselves. This is open to anyone over the age of 18. You don't need to have a dementia diagnosis to take part in some of these studies. So you can go to the Join Dementia Research website to sign up and find out more. And then finally, just to say a huge thank you to those of you that donate to Alzheimer's Research UK. All of the work we do is thank you to our wonderful supporters who fundraise, donate and volunteer for us, whether that's through holding bake sales, jumping out of planes, doing walks, doing marathons, giving money by direct debit, coming on to cheer at events, collecting donations, or speaking about the importance of research, and it all adds up. So it means that we can fund even more research. So huge thank you to all of you for your support this year. And if any of you feel like giving us a little Christmas donation, always welcome. But just to say thank you so much for all of your support. And then finally, just to talk about the other conversations we've had this year. This is the final event in this year's series of lab notes. We've talked this year about air pollution and its linked to dementia. We've talked about hearing loss, and we've been talking about to some of the scientists developing new drugs about the work they've been doing. All of those are available on our website to look at, to watch back on YouTube, as will this one. So If you've missed anything and you'd like to catch up, you can watch them all back by heading to our website or scanning the QR code. And that concludes tonight's event. So just want to say a huge thank you to both Paresh and Fiona, but also the team behind the scenes that have been answering the questions and fielding the calls and making sure everything runs over really well. So just to thank on behalf of everyone at Alzheimer's Research UK, thank you so much. Have a great Christmas, great festive season, and hope to see you in the new year. Good night.